Okay. Now, it turns out we also have facilitated diffusion channels that are voltage gated. In other words, they open and close in response to changes in membrane voltage. So we will put voltage gated sodium channels here. And over here we'll put voltage gated potassium channels here. Just put a couple. Okay. Ordinarily these channels are closed, but they will open if the membrane voltage changes. And that can lead to the firing off of a nerve impulse or action potential. Now I'm just going to erase part of this dendrite here to make a little diagram here. Here we have a little diagram here. We've got time on the x-axis, and we got voltage on the y-axis. And this time is, this whole space is just a matter of milliseconds, just to give you a little bit of perspective here. And voltage, membrane voltage. For nerve cells are ordinarily about minus 70 millivolts. Most cells are about minus 50. Here's minus 50. Here's 0. Let's say here's plus 50. Okay. This is the voltage across the membrane. So ordinarily the nerve cell is going to be hovering around 70 millivolts negative. A little more than typical for most cells, but not completely off the charts. Okay. Now what happens if a nerve cell gets an input from a dendrite, as we'll see later? What it means is ions are going to get into the cell and the membrane voltage will raise a little bit. So every time you get an impulse from a dendrite somewhere in that nerve cell, you get a little influx of ions and the membrane voltage might raise a little bit. But then it quickly drops back down for the most part. Now it turns out, remember we have the voltage-gated sodium and the voltage-gated potassium channels. Each of those two channels has a certain, what we call threshold voltage. A voltage, if you go beyond that, the channel opens up. It turns out that the sodium channel threshold, and we'll put this right here, is relatively low. You raise the membrane voltage a modest amount, and it causes all the sodium channels in the area to open up, and of course sodium rushes in. The potassium channels, on the other hand, have a much higher threshold. Okay, now let's see what happens to fire a nerve impulse. Our example here, we just had a little impulse from some distant dendrite, a few ions got in, and the membrane voltage for a few milliseconds went up a little. But you notice it's still below the threshold. But let's suppose we had several inputs at the same time, so you end up something like this. Here's one input, raises up a little bit before it drops back down. You get a second one, and drops a little bit. You get a third one, drops, and a fourth one, and we have just crossed the sodium threshold, sodium channel threshold. So what happens? These sodium channels open up. Sodium ions rush in. Now what's happening? Sodium ions are positively charged. We're allowing huge influx of positive charge into these cells. So what that means is the membrane voltage skyrockets, because now we're plunging positive charges into the thing. Okay, so it climbs up, and actually climbs up not only to 0, it even goes slightly positive. But look what happens. So we'll say sodium influx at this point. Sodium ions are rushing into the cell. And now our membrane voltage reaches the potassium channel threshold. Now the potassium channels open up and potassium ions rush out. At the same time, it turns out when you get the voltage that high, the sodium channels close back up again. They're only open over a limited range of membrane voltage. But now what's going to happen? Potassium ions rush in, meaning, or rather rush out, meaning we're depleting the cell of negative charge. So the potassium channels open up. Potassium rushes out of the cell, the membrane voltage plunges back down again. In fact, even a little bit lower than what it originally was. And folks, that is what we call the nerve impulse or action potential. Let me put that term down here. Action potential. That's a fancy word for saying nerve impulse. Okay. Now, how does this thing work? It turns out the very base of the axon, just where, yes?
What did you say the other name for action?
Nerve impulse. Okay. It turns out the very base of the axon, right where it joins into the cell body, that area has some modified channels that are touchier. They have a lower threshold than the normal ones. So an action potential or nerve impulse gets fired off at the base of the axon, and then it somehow propagates all the way down the axon. Now the way it works is this. In this little local example, we've just allowed sodium rushing in and eventually potassium rushing out. When sodium rushes in, you raise the membrane voltage, and some of these sodium ions start diffusing a little bit to the side, you know. Remember diffusion. They rush into this point, and then they start spreading. They start diffusing. And what that means now is enough of them diffuse over here, and these channels open up. So sodium goes in. Of course, the same thing's happening with the potassium, too. Ions diffuse in, rush into the cell, they diffuse outward laterally up and down the axon, and that causes these channels to open up. So more sodium comes in. So basically, if you've ever seen a sports event, what they call the wave, you ever seen that? You know, the person you're sitting next to raises their hands and you do the same, and the person next to you the other direction does the same. That's essentially what an action potential is like. Well, these go a lot faster, depending on the organism and stuff, but for the large mammalian neurons, that action potential goes at speeds that would probably win the Talladega 500. I mean, they're literally going at race car speeds, which is a good thing, meaning a nerve impulse in less than a tenth of a second will go from anywhere in your body and end up in your central nervous system. If you're a large animal like a whale or dinosaur, it may take, you know, several tens of milliseconds for the impulse to go all the way up, but it moves fast. Okay, now we're getting this action potential wave going down, and now we're going to go into the synaptic terminal and see what happens there. Well, the synaptic terminal has several things. One of them is just beneath the membrane, we have a whole bunch of membrane-bound storage compartments or vesicles. And these are filled with neurotransmitters. Neurotransmitters are the molecules that are going to be used to send a signal from one nerve cell to the next. So we have neurotransmitter vesicles. Okay, we got neurotransmitter vesicles. They're filled with neurotransmitters. There are many different kinds of neurotransmitters. Many of them are amino acids or modified amino acids. The thing called glutamate is just glutamic acid. It's an amino acid. Serotonin is a modification of another amino acid. Dopamine is a modification of the amino acid tyrosine. Acetylcholine is modified from another amino acid. So many neurotransmitters are amino acids or modified ones. Many other neurotransmitters are peptides, short chains of amino acids. Think of what they call the endorphins. These are the things that opiates bind to and activate the receptors. Endorphins are small peptides. Something called a nanodine. The receptors for that are stimulated by THC. In other words, these are the reefer receptors. Okay, those are peptides. So there are peptide neurotransmitters and amino acid or modified amino acid neurotransmitters. Each nerve cell produces and secretes only one neurotransmitter. So you can't make two or three at the same time. Okay, you only make one. Okay, now not only do we have these neurotransmitter vesicles lying just beneath the membrane of the synaptic terminal, we also have a third set of voltage-gated channels, and I'll represent these as kind of these weird triangles. And these are found only in the synaptic terminal. These are voltage-gated calcium channels. Ordinarily, the channels are closed. But when the membrane voltage rises, and that means when an action potential propagates into that synaptic terminal, when the membrane voltage rises, these calcium channels open up, and calcium ions now rush in to the synaptic terminal from, of course, high to low concentration. Turns out that calcium ions are going to cause these neurotransmitter vesicles, there's a protein that mediates this, to move towards the cell membrane, fuse with it, and dump their contents out. In other words, they're going to dump the neurotransmitters into the synapse. What I'm going to do is just draw a few stages in this process. Here we got some intact vesicles here, but now calcium comes in, we're starting to get vesicle fusion, and now the neurotransmitters are starting to leak out a little further on in the process. So now more neurotransmitters are leaking. So pretty soon we flood the synapse with these secreted neurotransmitters. Now once again, this process takes about a millisecond to happen. The synapse, of course, the cells of the one nerve cell is not physically joined to the next. They're separated, but they're only by about 20 nanometers. Those neurotransmitters in about 1/10 of a millisecond will diffuse all the way across the synapse. Remember, diffusion works really good at small scales and with small molecules. It doesn't work too good at large scales and large molecules. Okay, so now, in the dendrite, we have another set of ion channels. Facilitate diffusion again. These are ligand-gated ion channels, meaning they open up when a particular molecule binds the channel itself. And these are what we call the neurotransmitter receptors. I'm going to put that over here. The neurotransmitter receptors, once again, are ligand-gated. Facilitate diffusion ion channels. Okay, neurotransmitters bind to those receptors. It causes the channels to open up. So let's take a neurotransmitter right over here. Now the channel is open, and ions can either go in or go out. More on that in a minute. Okay. That influx or outflow of ions is going to eventually lead to the potential activation of this nerve cell here. Okay. Now, one or two other things I'd like to mention. It's okay if I erase at least part of this diagram here? Okay. Now, here again, each neurotransmitter will bind to only its particular receptor. So there are acetylcholine receptors that respond to acetylcholine, but they won't respond to any other neurotransmitter. There are dopamine receptors that respond to dopamine, but not other things. And there are anandine receptors that respond to this peptide neurotransmitter anandine, and just so happens that the nice little substance delta-9 THC in marijuana happens to bind to and activate those receptors just like anandine does. So that's why it still works. Okay, well, it turns out though, depending on what the receptor is, it's going to vary in what ions it lets in or out. It's all going to be facilitated diffusion, but there are some receptors there are some neurotransmitter receptors called excitatory receptors. And they're called that because what they do is when they open up, they're sodium channels. They let sodium ions rush into the cell, raising that dendrite's voltage across the membrane. And what that means is it tends to fire off that nerve. So an excitatory synapse with excitatory receptors is going to tend to fire the nerve cell that it's associated with. But there are also inhibitory ones. Most of the inhibitory ones, they do exactly the opposite. Most of them allow potassium ions to go out. So what's going to happen? You're allowing positively charged ions to go out of the cell. The cell is already negative, and you're losing more positive ions. It means the membrane voltage drops from -70 to maybe -80, even -100 in some cases. What that means is a lot less likely that that nerve cell is going to be fired off. So an inhibitory synapse means it's going to prevent, or at least try to prevent, that nerve cell from firing. Okay, so we'll inhibit that nerve cell firing. Okay, we got one or two more aspects of